So now to introduce our speaker, Susan Tian. Susan Tian is a microbiome researcher specializing in synthetic microbial communities and multi-omics analysis. She holds a bachelor's in biotechnology, where she graduated summa cum laude from Rochester Institute of Technology and is pursuing a PhD in microbiology here at Penn State. Her research on C. difficile inhibition, integrating computational and experimental approaches, has earned her a Paul Berg Prize in Molecular Biology and multiple presentation awards at national and international microbiome symposia. Susan is skilled in NGS data analysis, machine learning, notobiotic animal studies, and molecular biology techniques, and is also actively involved in mentoring, STEM outreach, and graduate leadership. And as a token of our appreciation, we would like to present to you this plaque for participating in the seminar.
Thanks for the very wonderful introduction. And then, so I actually have a natural very low voice. Let me know if you can't hear me, especially for the people at the back, and then I'm happy to repeat any of you guys. So first, welcome everyone, and thank you for showing up to listen to my talk. So I'll be starting to introducing my project for my PhD journey, and it's on how I crafted resistance to C. difficile infection using synthetic communities and using it as a tool to study the mechanism of action of actually how microbiome inhibits C. diff. So before I get there, I actually want to show this very typical figure of separating from correlation to causations. So in a very hot summer, we probably want to get more ice cream because we're hot. But for some reason, the homicide rate also increases. But we can say that ice cream sales increase are correlated with homicide rates increase, but that doesn't necessarily mean that one will cause the other. And we actually see that a lot in microbiome studies, where we see that there are differentially abundant microbes from different phenotypes of the microbiome. And I think it's a very essential step of actually moving from correlation to causation, of actually showing that those microbes have or have not any relationship in causing those diseases. And I think to be able to get there, one of the tools is synthetic microbial communities. So we can build the synthetic communities with the microbe that have different correlations to different phenotypes, and then perform, for example, multi-omics approaches or notobiotic models to test the correlations and to see if we can move from correlation to causation. And today I'll be using C. difficile infection as a model to exemplify this. So it's well studied in the literature that C. difficile infection is actually caused by antibiotics. And C. difficile infection is, C. difficile is a gram-positive anaerobe that can produce spores. And it's the spores that make it really hard to get rid of, which is also why it got its name, difficile. It means difficult. So they are reported by the CDC. There are around 500,000 cases of CDI every year, and it costs billions of dollars to treat. So it's definitely a problem. And so C. difficile is actually an opportunist pathogen, where when our microbiome is in a balanced state, it provides colonization resistance against C. difficile. And when the microbiome is being disrupted, usually by antibiotics, because notch is a very broad spectrum killing effect. So it not just kills the pathogen, but also the microbes that can provide those colonization resistance. And as a result, opportunist pathogen C. difficile can proliferate and produce toxins that can cause diarrhea and colon inflammation. So because C. difficile infection is caused by a bacterial pathogen, it's treated with more antibiotics, which brings the problem of a prolonged disruption to the microbiome. And very often cases, there will be recurrent infections of C. difficile. And so it's really this vicious cycle of antibiotic and more C. difficile infection. And FMT is actually a microbiome therapeutics that's approved by the FDA to treat RCDI. And it works by reversing this disruption by literally transplanting the fecal matter from a healthy individual to a recipient in order to restore their microbiome. However, there are actually a couple of problems associated with FMT. For example, to this day, we still don't have a definition for what is a healthy microbiome that is for everyone. And also, fecal microbiota transplantation is heavily reliant on human fecal donors. So, and then our microbiome changes very rapidly. So, which makes it really hard to reproduce or generate it in large scale. And more importantly, we don't really understand how FMT works. There's a lot of hypotheses. It might be secondary bile acid, it might be short-chain fatty acid, but we still don't have the one hypothesis that is proven. And actually, it can be risky as well. So FMT can actually as a vehicle to transmit pathogens. So there are, in 2019, it's reported by the FDA, there are 6 cases of FMT recipients got infected with enterohemorrhagic and enteropathogenic E. coli. And 2 of them actually died from the infection itself and not the disease that they already had. So this leads to my research question is, can we use a synthetic microbial community as a replacement to FMT to treat C. difficile infection that is more reproducible and safer? And also, can we use synthetic communities to understand how FMT actually works? So I started with doing a meta-analysis. So the goal is identify the microbe that are anti-correlate with C. difficile that should go into the synthetic community. And also, meta-analysis approach is more conclusive than analyzing single studies. So from PubMed research, I went from around 300 data sets to 12 data sets. And this is because a lot of the data set either don't have relevant study design, or they don't have sequencing data, or they don't have the metadata that is suitable for analysis. But that still gives me a total of 900 samples, which is bigger than any of the single studies that I have. So in this figure, it reports the analysis of the meta-analysis. In each row, it represents the analysis of single studies, whereas at the bottom, where it says combined, it means that all the study sets are combined into one data set. As you can see, when C. difficile abundance goes up, the richness of the microbiome actually decreases. And this is probably suggesting when C. difficile is colonized, there is a loss of certain taxa from the microbiome. And next, when I compare the microbiome samples on an ordination plot, so where each dot represents a microbiome sample, that C. difficile colonized microbiomes are clearly distinct from the microbiome from healthy controls. So this is showing that not just that the microbiome has shifted when C. difficile is colonized, but more importantly, if we can use this difference in the microbiome to predict C. difficile status. So I then fed the meta-analysis sequencing data into a random forest model, and I used the leave one study set out approach, so that every time when I test the model with the data, it's seeing a new data set every time to increase the robustness of the model. And I also excluded C. difficile from the data set. This is to see how well the model can predict C. diff just using the rest of the members in there. And this is the receiver operative curves. So the middle line represents the predictions are all random. There's a 50% true positive, 50% false positive. Whereas the closer to this corner, it means that the model is more robust. And looking at the area under the receiver operative curve numbers, it's showing that the prediction is doing pretty well of using microbiome composition to predict C. diff. And another reason that I use random forest, other than it works really well with noisy data, sequence data will be one of it, is that random forest can actually provide features importance that goes into the prediction. So in this figure, where it's showing the feature importance over the number of ranks of the features, we took the top 200 most predicted features to work with the downstream analysis. And first is because it's a workable scale, and also the features after that seems to have diminishing return in its predictive strength. So even though including it, but it might not really help with the prediction. So from there, these are the abundance of the top 200 most predicted features. And as you can see, for the negative well, first of all, they do cover a pretty wide range of taxonomic units. And the negative predictors, most of them seems to have a decreasing abundance, and the positive predictors seems to have a increase in their abundance. And this further suggests that maybe the replenishment of these negative predictors may be the key to study how the colonization resistance happens. And the next thing I do is to study the correlation calculate the correlation of each feature to C. difficile. So in this figure, where C. difficile in the center and each feature are represented in the circle, the blue lines represents there's a negative correlation, positive, red lines represent there's a positive correlation. So I want to focus your attention on this cluster of features here, that out of the negative correlators, there are a group of features that positively correlate with themselves. And this probably suggests that when these members are cultured together, they're probably going to form a pretty stable network. And I also use this as the basis of my two synthetic communities, SFMT1 made with negative correlators and ProCD made with positive correlators. And before I move on to more experimental data, I want to point out that C-sendence did not came up in the prediction. And the importance of this information is that C-sendence is well studied in the literature, that it can convert host-produced primary bile acids into microbial produced secondary bile acids that are directly inhibitory to C. difficile spore germination and outgrowth. So I then manually include C-sendence into a variant of my synthetic community to test the role of secondary bile acid conversion in C. difficile inhibition. So from the features, I aligned the 16S RNA genes to the lab strain collection we have. And from there, we also removed the redundant features. And there are also some features, some strains that we just don't have in our lab collection. So that get it to around 130 features to 37 strains in SFMT1 and to 25 strains into ProCD. And so how did I put a community was that I grow them one by one and verify their identity by senior sequencing to make sure that I didn't bring anything to contaminate the rest of the community. And after that, I put them by a fixed number of optical density. So they should be relatively averaged out in terms of their abundance. And I also included a human FMT as a clinical standard to compare. And the first thing I look at is how their stability change over time. So this community in vitro is pretty stable for the course of 4 days, whereas it might be more stabler than a human FMT. This is probably suggesting that like we still can't culture all of the members from a human FMT. And next, we move on to look at the stability over time and the metabolic functions of synthetic communities using germ-free models, where we colonize the germ-free mouse with for 14 days, and we measure the total bacterial load, 16S RNA sequencing to measure the compositional changes, and metagenomic sequencing to have higher resolution to the strain level, and bile acid and short-chain fatty acid to look at the metabolic functions. So first, in terms of the total bacterial load colonized in germ-free mouse, we found that this community can stabilize after 3 days of colonization. And that seems to be true from the sequencing data as well. And and next, these are the maturation or the colonization patterns of these strains. So first of all, we have the controls, which are the communities themselves. They are put relatively averaged. And second of all, in vitro colonization models does differ from in vivo colonization models, and this is probably because the microbes are living in a very different environment, whereas in vitro the environments are more fixed, but in vivo is more dynamics. And in vitro, communities are dominant by E. coli and some other bacteroides that have higher tolerance to oxygen. And in vivo, it also started with, for example, facultative anaerobe E. coli, and it's probably consuming the oxygen and makes it more suitable for the strains that are more oxygen sensitive to colonize in the later time points. And we do see that pattern as well. Even though not all of the members do colonize, and this is probably a function of their oxygen sensitivity because these are from glycerol stock to a syringe and to animals. So there does got some chances to expose to oxygen. And also, there might be also competition going on between the strains. And we do see that when we perform metagenomic sequencing to look at the strain levels. And we found that for each strain, for the strains within each species, there are competition going on where there's a dominant strain per species. There's also the less dominant one. This also showcased the possibility of using synthetic communities to study microbial ecology as well. And from there, we look at more of the metabolic functions of the community. The first thing we assayed is the ability to generate short-chain fatty acid, which are primarily a microbial metabolite. So compared to the germ-free control, SFMT1 does significantly increase the production of short-chain fatty acid, and then it also were able to produce more acetate and propionate than human FMT and less butyrate. And this is probably the function of what's in there and what's in the human FMT. And it does capture most of the metabolites that a human FMT can produce. Next, we look at bile acids, and shout out to Min-Soo who run the samples for me. And first, we look at total bile acid production, that these two communities does produce a comparable amount of total bile acids than the human FMT. And in terms of secondary bile acids, it does produce less compared to human FMT. And so this is more of a matter, I do like to point out, we were just only using one human samples in this experiment. So depending on the composition difference in another fecal sample, this might look different. And we then to the more important question is, what does C-synthase actually do? And then we were able to show that the inclusion of C-synthase compared to the community does not have it, does increase the production of the typical secondary bile acids, LCA and DCA. And next, by comparing their the composition of their bile acids, it does distinct from human FMT, but actually this could be a potential for understanding how to improve the performance of synthetic communities and to go from there. So next is to the question of how, back to the goal of why we designed these communities to see if it can actually inhibit C. diff. So we co-cultured different communities with C-synthase in vitro, and we found that pro-CD does not really inhibit or make C. diff cell grow better compared to the media control, but the two communities was able to reduce the growth of C. diff cell by orders of magnitudes. So this was a very hopeful preliminary data for us to move on to animal models. we did shrink the colonization days to 7 days after we found out that the community can become a stable community after 3 days of colonization. And from there, this is the weight loss over time after the infection period. So the two communities were significantly able to protect the animals from the infection-induced weight loss compared to the germ-free control, but is not comparable to human community. And that is also true in toxin production. And this actually, it's a motivation for us to really understand how human FMT works in order to improve the performance of the genetic communities. And also another conclusion for this piece of data is that the addition of C-syndens does not really change the performance of how it behaves on inhibiting C. diff. So the bile acid conversion might not be the major driver for the inhibition. And we also use a more clinically relevant animal model where we try to mimic the infection process might look like in actual human, I guess, patients. So we started with treating the conventional animals with cefoperazone, which is an antibiotic, and the goal is to lower the colonization resistance of the microbiome. And after washout, we introduced spores to the animals to start the infection. And we also treat the animals with vancomycin, so this really mimics how the clinical process will look like. And then we give the animals 2 doses of FMT to study how well they can prevent recurrent CDI. And we found that even though SFMT is very superior, it's superior to the mouse FMT in terms of preventing infection-associated death in these animals. But I do like to point out that we also found a considerable amount of vancomycin-resistant enterococcus, the results of vancomycin in our model. And the problem is that it really masks the function of genetic community in actually treating C. difficile infection. So for that reason, we have to go back to the germ-free model. So now is the question of what is the mechanism of action of the communities or of FMT that can lead us to design for a better treatment. So we went back to the metabolic functions, which is quantified by NMR of the genetic communities, and we found that proline was significantly depleted by the community, and its fermentation product 5-amino valerate was enriched. And that's the only pair that we found that have this pattern. And this is the pathway. And the importance of this is that proline fermentation is the major energy intake pathway for C. difficile. And it was shown in another group that when C. difficile was grown or was infected in a healthy germ-free mice versus a dysbiotic germ-free mice, and we found that first, so there is colonization resistance of the healthy microbiota, and also they also compared a wild-type C. difficile to a PRDB mutant of C. difficile, which so PRDB is responsible for performing stickling fermentation proline. So this shows that when C. difficile lost the ability to perform stickling proline fermentation, it does have lower colonization and also lower toxin production. So this further test shows that proline is a very important nutrient for C. difficile. So we wonder, is any members that came from the genetic community that is limiting the proline as a way to inhibit C. diff? So the first thing we do is to identify the members that have the proline reductase homolog. There's 8 of them, and when we took the A members out from the synthetic communities, we saw that it completely abolished its ability to convert proline into 5-amino valerate. And in fact, it actually liberates more proline that can potentially feed C. diff. And we further quantified this by measuring the proline consumption and 5-amino valerate production in each of those 8 strains. We found that there is just 2 members, is 29 and then 254, which are Doria longivitina and Papostreptococcus anaerobias, that are very potent at performing this pathway. And we then make another variant with just these 2 members to test the function of proline fermentation, and also a knockout community to compare the loss of this function in inhibiting C. diff. And using the same germ-free model, we found that when the proline fermenters are taken out, this light purple here, the rest of community or the knockout community lost its function of providing the protection, whereas the 2-member community is sufficient to provide the protection. And then we cross-reference to the clinical data from the meta-analysis. We found that it's indeed these 2 organisms present in higher abundance in C. difficile negative samples than in C. difficile positive samples. And as we further quantified the colonization of these 2 organisms to study their dynamics, we found that it's really a monocolonization of P. anaerobias. So we proceeded with a monocolonization, and we found the same, that proline fermenter, this 1 strain, is necessary, means that when it's taken out, it no longer provides protection and sufficient to provide the protection. And it even performs comparably to a human FMT, and especially on 6 days post-infection, the P. anaerobias is able to reduce more toxin production than the human FMT. We further quantified this. We further quantified the inflammation severity by doing Luminex assay, and this was done by Sunshine in Vishal's group. And that we found that indeed P. anaerobias was able to lower the production of pro-inflammatory cytokines such as IL-1 beta, IL-6, and TNF alpha. And so at that point, we were wondering, how do we show that proline fermentation is the only mechanism and not just other mechanisms that can provide the inhibition? So the first thing we did is actually gave the proline back to reverse the competition and to see if that can reverse C. difficile infection. But we tried it with supplementing the animals with proline in water or in diet, but we failed to increase the cholinic proline concentration. So that forced us to have to explore this in vitro. And so before I get there, I want to provide a little bit more background on C. difficile life cycle, where it starts from spores, and when the spores sense there is a germination trigger, for example, primary bile acids, it will germinate and goes into the vegetative form, and that produce toxins and more spores. So in order to understand how can we target this, the first thing we look at is if the fermentation pathway itself or the product of it is inhibitory to spore germination. And we compare 5-amino valerate, and a note is that we were using a biological relevant concentration for this assay. And when we compare the 5-amino valerate effect to taurocolic acid, which is a known inducer for germination, and CDCA, an inhibitor for C. diff spore germination, we found that 5-amino valerate does not act as either an inducer or an inhibitor. And next, we look at how does it affect vegetative growth. That it does not really change the growth of C. diff cell that much, and it's the same for toxin production, that it has no effect. So from there, we go on to the next hypothesis is to test if P. anaerobias is inhibiting C. diff through a contact-dependent manner. So in this assay, we spread C. difficile on an agar plate, and then we load two P. anaerobias on top of it so that they are directly contacting each other. And then we were measuring zone of inhibition that we found there's no inhibition by direct contact by 254. And we were using a variant of this assay called defer antagonism assay, where this is just a plate of the positive control shown here. So we first streak the bacteria horizontally and grow it for 24 hours, and then we remove the physical bacteria. But whatever the bacteria produce will stay in the agar. And then we cross-streak the acid bacteria to see if the metabolite will do anything to the bacteria on top. So this is my positive control, Lactobacillus reuteri, and it has shown before that this strain will produce reuteri that will kill C. diff. And that is true that we see zone inhibition here. And this is a plate of P. anaerobias that we found that there's no zone inhibition by 254 to C. difficile. So this shows that a P. anaerobias does not either produce an antimicrobial compound or it's inhibiting C. diff through a contact-dependent manner. And next, we look at bile acid. So we found that even though P. anaerobias was able to provide the full course protection compared to the germ-free control, but it does not produce any bile acid that were differentially abundant. And next, we're trying to figure out what exactly P. anaerobias can consume. So we perform NMR to quantify the metabolites in the supernatant of P. anaerobias. Compared to the media control, we found that it's able to consume 6 different amino acids, and these are the 6 correspondent fermentation product. And we were wondering if we could use the supernatant because it was already depleted for its nutrient, if we could use it as a minimal media so that we can add in one thing at a time to decide which is actually the key of limiting C. diff. So this is, again, is the control where we grow C. diff in the supernatant. It does not grow. And when we add BHIS, which is the media, back to the supernatant, C. difficile can grow. Means that the supernatant is a very nutrient-free context, whereas adding the media back can bring in some of the nutrients that was depleted. And then we add in glucose and the essential vitamins and minerals. So this is just also glucose as a carbohydrate source, and C. diff also couldn't grow. Only when we add in either straight form of proline or a mixed form of proline from peptone that we can see proline is able to salvage the growth of C. difficile in this consumed media. And we further verify this by engineering proline utilization A gene, which is a different pathway but still uses proline to convert glutamate. And we found, compared to the vector control, that the strain that's expressing proline utilization A gene, it was able to inhibit C. diff in vitro. So from there, I would really like to conclude that the meta-analysis, it's a very reliable approach to study, to design the synthetic community with certain functions, and also by using synthetic communities, we were able to find that bile acid production or bile acid conversion is not the major driver, whereas proline limitation in the environment is more important for C. difficile limitation. And also, hopefully, I was able to show that by leveraging the composition of the synthetic communities, we were able to test different functions in a complex community in other disease pathogenesis. So I really like to compare this to a biting the needle in the haystack, where we went from over 10,000 OTUs to one strain and one metabolic pathway. But also this also showcased the ability of what synthetic communities can do. So the current literature and the technology we have, it's pretty informative of who is in the microbiome in different phenotypes. However, there's still the rest of five answers remains to be, or five questions remain to be answered. For example, how does it happen? And what are they doing? What are microbes in different phenotypes are doing? And why? So what caused the differential abundant? And when to where? So this is probably context dependent. For example, we might worry about when we think about early life exposure to these microbes, and also where. For example, where geographical locations matters. For example, IBD. And not just to a human disease, but synthetic communities can also be used to answer these questions in agriculture or to plant, to study how plants behave or how plant talk to their microbes and the two bidirectional relationship, and also some other cases. So with that, and then I would like to take any questions.
Thank you, Susan. Fantastic talk. Traditionally, our first question will come from a trainee. So if you are a trainee in the microbiome seminar or a series, please feel free.
Go ahead.
Hi, great presentation. I work with synthetic communities, but more so in plant systems, so I'm not sure how the animal systems work. Can you just let me know how are you putting these synthetic communities into notobiotic mouse?
Yes, so we were doing the oral gavage approach. So where it's using a curved needle with a ball tip, and then you connect it to a syringe, and then you load the bacteria in there, and then you push it through your mouth to get them to the stomach first, and then it moves into the GI tract. I've also read online there are other methods, for example, going from the rectal directions. There's also other it's also a feasible method to do it.
Hi, fantastic project. It's very thorough and compelling. And I love that the initials of your magical microbe is PA for Pennsylvania. Of course. So let's call it the Pennsylvania microbe. Can you remind me again where that sat on the feature importance list when you did your initial analysis of machine learning analysis? Was it intermediate, high, low?
So it's actually more of towards the intermediate to low level.
Yeah.
The top predictors are actually, well, C. diff itself and E. coli and Faecalibacterium bacteria.
Did you have any sense of maybe think about why it landed at the intermediate level rather than?
Yeah, so I think first of all, the data type that we fed it was mostly abundance, or like their composition and their abundance. So I think if we couple it with metabolic data, it probably is going to use that as a different or as an additional information to call for predictions. So,
Great. And if you did that, would you expect that there would be many more microbes with that same metabolic capacity that could inhibit C. diff?
yeah, I think yes, because I think it's well, first of all, not more data always necessarily means higher predictive strains, but I think in this case, in the C. diff cell model, whereas metabolic does play a role, I think that would
You know what you're looking for. So in theory, there could now be a portfolio of bacteria that might be able to do this. Okay, thanks.
Trainee, yeah.
Hi, so I work with bacteriocins and gram positives, and I was wondering if you have tried other media, because like in my case, I started with one medium and it didn't work for all my strains. When I changed to another that has asparagine, I recovered more activity on my plates, and also I use double layer agar and incubate for 4 days.
Yeah, so I think the media that we use are all that we know that it was able to support both the growth of or optimal growth of P. anaerobius and C. diff. So that was and then in cases where when the strains have different metabolic requirements, you probably need to use different medias, but I do think that
And did you add the proline to the media?
that already have, so it's media is primarily made with brain and heart tissues ground up, so it's very rich in amino acids, including protein as well. And if you remember at my NMR data, that does show that protein was abundant in present in a very abundant concentration.
Vishal first.
Yeah, sure. Thank you. You may guess I will have a question nutrition related. So I'm just coming back to first thing is a very great presentation and it's a very good story. So my question is like I think you were involved like your protection inhibition to the C. diff is much more related with the protein amino acid metabolism. So if you think like your strain of interest, whether it is sensitive to the like amino acid ratio, like if you change the amino acid composition, do you feel like those strain level will go down or up? So in other word, I'm trying to ask a question like if the protein environment shift or amino acid change, so whether you expect the similar level of inhibition or
Yes, so that's a very good question. Essentially, we have tried to answer that question by doing the water and diet supplementation, where we also had a group where there's zero amount of protein. But I guess P. anaerobius is very efficient at liberating protein from their diet, so it's really hard for us to from that part. But I do think that if it's worth of trying if we that co-culture P. anaerobius and C. diff at when there's no protein around to see if the competition is still offered.
I'm gonna add, because my question is the same. Did you try to look at the speed of degradation or uptake of proline when you just do the media and 1 microbe at a time for P. anaerobius versus C. diff? Because we do that.
I didn't do it for those strains, but I did it for my mutants, and then it does show that so we first assay that if the strain with the part A can convert from proline to glutamate first before we move on to that state, and that it does increase the consumption of protein with the extra protein utilization gene, and then we move on to co-culturing back to C.
Because it basically just taking up all the proline, and then there's none for C. diff.
C. diff, yes, C. diff on itself cannot liberate protein from the diet, so it has to rely on other members of the microbiome that do that. Whereas if P. anaerobius in this case is very efficient at doing that itself and using the protein, so I think it's this case is a
Yeah, I was thinking about PA in the context of like a quote unquote like healthy or natural gut microbiome. Do we know where PA is coming from? Like, is it associated with like high fiber foods or, you know, things like that, like a source?
So it's a very good question, because actually in the literature, P. anaerobius, it's more associated with colorectal cancer. Which,
You're
yes,
not gonna
which,
see this, but
yes, which opens, well, but again, it's correlation.
yeah,
So it's
not causation.
and also, which also opens up a lot of areas in our lab trying to use other microbes and design live biotherapeutics with them instead of P. anaerobius.
Jane?
I guess mine is kind of, my question was building on that of taking this back into like the FMT context. What would you see as kind of the next step? Is it checking that if we're doing FMT treatments, we make sure that they have a large part of P. anaerobius, or just trying to augment with that alone to see if that works in like in humans at this point?
I do see multiple lines from what yours just said. So first of all, I think when it gets to FMT, there is an essential question of engraftment that goes with it. And also FMT, like I mentioned, it's really hard to reproduce the composition. So depending on that, we might carefully design the experiment to make sure that we're comparing the colonization of P. anaerobius with representative features from the fecal microbiota. And so those are the questions, or those that are what I'm thinking of.
I have a question when you were mentioning about advantages of synthetic community over FMT. One of the things you mentioned was like there have been instances of infection in people who receive the FMT. Has it already happened, or don't they sequence what kind of communities are they inoculating in the recipient? And don't they do sequence for potential pathogens?
Yes. So I think especially after the where the infection case is reported, there is like more prestigious methods of screening for pathogens and other, not just microbial pathogens, but virus and other stuff too from the fecal sample. But there is another layer of to that question is we don't really know if the same microbe going to be act as a healthy microbe in my gut than in your gut, right? So that's also a essential question remain in FMT. But if we are using a microbe that we know are safe, for example, like or collanisol, to design for life biotherapeutics, it might be already starting at a safer standpoint, right?
So one of the things that you mentioned is maybe there's going to be some like contact dependent inhibition. And this is kind of a tangential question, but do you know if like a lot of bacteriophages infect C. difficile in the gut? That could be like a
I've actually
useful.
read about it. There's like phages and then people develop that into use as like a therapeutic source. But I think with phage, the question remains how fast that C. diff will come over to develop resistance to components.
Sure. So I had one more question. If no one else has any questions. I was interested going back to how you determined, if you could repeat, how did you determine that your SynCom strains were competing with one another? That was a little bit.
Sorry, I didn't hear the second.
How did you determine that you had some strains in your synthetic community that were competing with each other?
So one was that there's the metagenomic data was showing that within the species there are strain level competition. But so we haven't really assayed like other microbes if they are going to compete or not. Yes, which is mostly on the strains. Because they probably have a very similar metabolic niche. So that means that they're probably fighting for the same set of nutrients, and depending on if you're faster or I'm faster, it's going to be a competition.
All right, let's thank Susan for